All right, I'm going to do a quick introduction to this third and final panel. We've had a great two panels already at the University of Florida Diabetes Institute, and we're very, very pleased to have us or have with us three scientists and a physician scientist actually, and along with Mark Atkinson, who for a long time was the director at UFDI and also one of the founding fathers of the NPOD research network and repository, which is so important to all the work being done in T1D and especially at UFDI. Let's see a little bit about the scientist speaking today. Dr. Maigan Brusko is an assistant professor at the University of Florida Diabetes Institute. Her academic career has been focused on cellular immunity in clinical subjects and with translational research and clinical trial monitoring, preclinical biomaterial therapeutic testing, and single-cell biology in cell therapy approaches, sepsis, and autoimmunity. Her role as scientific director of the Human Atlas of Neonatal Development and Early Life Immunity, or HANDLE-I program, has focused on the understanding of immune architecture and cellular maturation in immune tissues, particularly the thymus, in context of donor age and immunogenetics. Brittany Bruggerman, MD, PhD, is a pediatric endocrinologist and physician scientist, and she's been involved in T1D research pursuits for 15 years. And then Dr. MacKenzie Williams, she's a PhD. Her role within the University of Florida Diabetes Institute followed a unique evolution. First, she was a friend of the lab, working as a barista, and then a student under the mentorship of Mark Atkinson, followed by a postdoc in Clive Wasserfall's lab. And she's a newly minted faculty member within the Wasserfall lab. She's furthering our understanding of the pathological mechanisms that lead to beta cell dysfunction and assessing whether serological markers such as exocrine pancreas enzymes, C-peptide, et cetera, can be used to predict insulitis, low relative pancreas weight, and other hallmark features of type 1 diabetes. The topic of this panel is exploring the whole pancreas in type 1 diabetes. And I didn't know, Mark, if you'd like to set the stage on this. I think it would be very helpful if you would.
Absolutely. Thanks, Monica. And again, on behalf of everybody at UFDI, we greatly appreciate your setting this up for us today. And again, kind of as you said, I'm old, as you inferred, but last August, I hit my 40th year in type 1 research and care. And I'll have to say that when I was putting this brief introductory talk together today, I was thinking about why care or who cares. So I ended up talking about who cares. And the reason is as follows. If you go to the next slide, I actually tried to act like I was young, and I asked ChatGTP a question as into what do we really know about how type 1 diabetes develops? And it came back with this answer that the statement type 1 diabetes results from the autoimmune destruction of insulin-producing beta cells is widely accepted and frequently cited in the medical literature. And I've been saying this for a long time, and I'm glad ChatGPT backs it up, is that if you look at articles on the pathogenesis and natural history, this is almost the intro to every article or some variation thereof. And there's a lot of good reason for that. And it has to do with what we perceive for what I've actually goes back not just 40 years, but 50 years about type 1 diabetes and autoimmune disease. And for much of the time, it's rained down on this principle that somehow there's a loss of tolerance. And again, as we began to understand differences in central and peripheral tolerance, that we don't respond well to beta cell antigens. And then this leads to beta cell destruction. And then on the next slide, if you show this, again, this is just an example in 3D of a case of NPOD that we'll mention in a minute, showing insulitis. So in here is an islet stained in purple for insulin. And with the CD8 cells, this is from a recent onset type 1 diabetes patient. You can see that there's infiltrate of the islet and the surrounding tissue. So I'm not here today to question that type 1 diabetes is an autoimmune disease. But what I am, and my colleagues here are come to say, is that there's more to the disease than just autoimmunity. There's the role of the beta cells that you've heard earlier on, plus the third component, and that is the pancreas. Next slide, please. And so, again, it's been mentioned all throughout the talks today of this effort called NPOD that, again, was started back conceptually in 2006, and then in 2007 we actually got going. But it tried to take a look at how can we go back and mine essentially the human pancreas to make discoveries like we have done throughout history, from studies in the 1800s noting the islets of Langerhans to Schmidt, a German pathologist in the early 1900s that was the first to observe the insulitis infiltrate that I just showed it, but in a different form. And then other investigators that began to notice that people with type 1 diabetes not only had two forms, non-insulin requiring and insulin requiring, but depending on the age you were diagnosed, that the pathology looked different. And then with that, we went and started NPOD again with its official start in 2007. Next slide, please. And what did we learn? So we were literally about 4 or 5 years into the program, and the technicians that were under the hood, as you would say, came to me and said, we're noticing something different. And that was that the pancreas of individuals with type 1 diabetes were typically smaller than those that did not have the disease. And, you know, it started us to take a look at it. And we published our first paper on this on the right a little over a decade ago in JAMA, where we noted that if you compared age and eventually BMI matched individuals with type 1 diabetes versus controls, or even those with autoantibody positives, that there was a difference in the weight. Now, for full credit, there were a couple of studies in the 1950s and 60s that were lost that noted this, but again, this was before we even had the definitions of type 1 and type 2 diabetes. So it was led to confusion. And where are we today? And this is what my, next slide, please. This is what my colleagues are going to talk about in their talks, is that we now know that there is pancreas involvement or relationship between it and changes in the pancreas. And you'll hear a lot about size, change in histology, and then even on the right-hand side is a recent scanning electron microscope image of what we've done from the pancreas of somebody with diabetes, and taking a look at changes that occur in either enzyme production, pancreas weight, and others. And so I commend, and it's not just University of Florida work, there's people all around the world that are looking at this. And it is really an interesting area outside the norm, but we think that pancreas contributes or is influenced in type 1 diabetes. And I look forward to the rest of the conversation. Thank you.
Thank you, Mark. That was great. Great way to set the stage. And now I'd like to invite, I think, Dr. Bruggerman to walk through her slides with us.
All right. Yes. So I'm Brittany Bruggerman. I am an MD, as was mentioned. So I bring a little bit more of a clinical perspective to the whole pancreas and type 1 diabetes. So Mark went over this, but I always think it's really interesting. This is the typical timeline that we all know well in terms of the milestones of type 1 diabetes discovery. So 1921, we have the discovery of insulin. In 1974, we have the discovery that autoimmunity was the cause of type 1 diabetes. In 1986, we had the first immune therapy trial. And then another big milestone that I've been excited to be a part of is in 2022, we had the first FDA-approved disease-modifying therapy. But what often gets left out of this timeline is that we actually knew that the exocrine pancreas was affected in type 1 way back in 1909. So one of the first studies showed that there was decreased pancreas size way back then, and interacinar inflammation. And then in 1925, patients with exocrine dysfunction were described in type 1 who had type 1 diabetes, so 100 years ago. But there was this kind of big lag because, as Mark was describing, we didn't really understand the classification of type 1. We really focused in on the islets once we discovered the autoimmunity component. And so the exocrine pancreas was kind of ignored for a long period of time because we thought, well, maybe it's impacted, but not as relevant as the islets. But in 2012, work by the UFDI did show that the pancreas weighs 25% less in autoantibody positive non-diabetic organ donors. And that really revitalized the field of looking at the exocrine pancreas in type 1. Again, as a clinician, just to go over briefly normal exocrine physiology. So the islets comprise only 1 to 2% of the pancreas. And the exocrine tissue, which is mainly acinar cells, comprises 95%. And so this reduction in size really is largely due to a decrease either cell size or number of acinar cells. Acinar cells form acini and produce and store digestive proenzymes. So such as proteases, trypsinogen, elastase, and chemotrypsin, amylase, lipase. And these are released in response to CCK and vagal stimulation after a meal. After a meal, the change in duodenal pH from stomach acid being delivered and secretin causes stimulation of ductal cells to release large amounts of sodium chloride, bicarbonate, and water via aquaporin channels and CFTR channels. And these have also been shown to be, CFTR at least has been shown to be affected in type 1. And it can be, a large amount. So 1.5 liters a day of this fluid is produced typically. And this helps with digestion of our food. Another thing that we've discovered recently is that there is actually a large amount of endocrine and exocrine crosstalk. And so this does make sense because they come from the same progenitor and they're both working together to digest and utilize nutrients. But we just, again, focused in on the islets for so long that we didn't really explore the crosstalk between the exocrine and endocrine pancreas as much. One recent study showed that carboxyl ester lipase influences beta cell growth and function. And then a really, really interesting study showed that MODY8, which is a genetic form of diabetes, causes mutant cell protein to aggregate within beta cells, and that is the cause of MODY8. So again, an exocrine cause of diabetes there. It also goes the other way. So inflow, there's a really interesting work on vascularization of the pancreas going on as well, but the inflow acinar portal vascular system exists. And so even though islets only comprise 1 to 2% of the pancreas, they get 20% of the blood flow. And then that blood bathes the rest of the exocrine tissue and the venules that exit the islets. And this leads to acinar cell growth, amylase synthesis, and secretion. And so the insulin does influence the exocrine pancreas. Glucagon also influences pancreatic enzyme and juice secretion. And chronic glucagon exposure can cause degranulation and atrophy of acinar cells, which is, in type 1, we do have higher, chronic glucagon exposure. So what does this mean for the development of type 1? Because we're all interested in type 1 prediction and development and obviously prevention. And so in this sort of stage 0 period, is there a difference in the exocrine pancreas in those who are at risk, high risk HLA or first degree relatives, but don't have any antibodies, islet antibodies yet? So another study by the UFDI showed in 2019 that the relative pancreas volume is lower in type 1 diabetes first degree relatives who do not have islet autoantibodies. And then a study in India, Penno, in 2020 showed that in some progressors to type 1 or to islet autoantibody positivity, fecal elastase, which is the most common clinical marker of exocrine dysfunction, declines prior to the onset of islet autoantibodies. However, when they did a cross-sectional analysis of baseline differences, there were none that they found in that study by either progressor status or HLA. And then Giovenzana in TrialNet in 2022 showed that there are reduced amylase and lipase levels in first degree relative adults with type 1, but not in children. And these again are that don't have islet autoantibodies. This area has some conflicting research, and so other studies have shown no difference in exocrine enzymes, fecal elastase levels, or pancreas volume at this stage. So I think this is an evolving area that we definitely need more discovery in. There is more consensus that in stage 1 type 1 diabetes, there are changes to the exocrine pancreas. And so again, that same study in 2019 showed that in multiple islet autoantibody positive relatives, there was reduced pancreatic volume even more than in first degree relatives. Then another study by the UFDI showed that lipase and trypsinogen are reduced in multiple islet autoantibody positive individuals, which we'll get into more a little bit later. And this is some unpublished data that we had. I had done an analysis within NPOD donors who were younger than 30 because chronic pancreatitis is very common, more in older individuals. And so I wanted to limit that confounder. And so we looked at just individuals under the age of 30 in type 1 diabetes, multiple antibody positive, singles, and controls, and looked for pathology differences. And we did find that there is more acinar atrophy and chronic pancreatitis in multiple autoantibody positive donors within NPOD. In stage 2, there really is not any work yet looking at the difference in exocrine function, size, or any markers in between stage 1 and stage 2. And so that's one study that we are conducting right now. So I have basically in the TEDdy population. So this is the largest birth cohort study evaluating the pre-type 1 diabetes period. And we have banked stool samples, thankfully, from this study. And so we are doing a case control study looking at cohort A are people who went on to develop stage 3 type 1 diabetes within a set period of time. And cohort B are people who went on to develop stage 1, but then went a very long time without developing stage 2 or 3. And so we're comparing these two populations to see if exocrine function in terms of fecal elastase declines more at the kind of onset of islet autoantibodies or more at the onset of dysglycemia. And that's what we're trying to determine with this. And so this will not only help us to understand more about the natural history of exocrine dysfunction, but will also help us to determine if if fecal elastase particularly is a useful biomarker for progression of type 1 diabetes.
Can I just interject right there? Where are you guys in that study? Are you?
Yeah, we have basically finished the stool analysis. So we had to analyze 4,500 stool samples. And so it was a lot of ELISAs. So we're done, I would say, with 95% of the actual analysis of the stool samples, but we're currently blinded to subjects. But as soon as we finish up, we'll unblind and go ahead and analyze the results.
Exciting. Everyone should be on the lookout for that paper. Exciting.
Yeah, and I'm hoping to get it out sometime in the next year for sure.
Great. We'll keep an eye out.
Yeah. And then again, this is more established understanding that in new onset stage 3 type 1, there definitely is an impact to the exocrine pancreas. So people with stage 3 at onset typically have a 20 to 50% reduced pancreas volume and exocrine function, which people don't really realize that is present because the exocrine pancreas has a lot of redundancy. And so you can have a substantial reduction in amylase and lipase and fecal elastase and still have relatively normal digestion. However, there are a lot of people with type 1 who have symptoms that could be consistent with exocrine dysfunction who we maybe just aren't catching. And so there was a recent systematic review showing that in up to a third of patients with type 1, they may actually have clinical exocrine insufficiency and we just aren't catching it. So I'm also doing some extra research in that space that didn't really have time to fit into this talk, but we are looking at the clinical impact of some of this reduction in exocrine function.
It seems like that maybe there's room for understanding whether or not those patients who are undiagnosed with type 1, but in stage 1 or 2 identified, maybe there's room for some kind of enzymatic supplementation.
Yeah, it's interesting. It basically, your enzymes have to be quite low for you to have clinical symptoms is our understanding at this time. And so in the pre-type 1 period, I don't think right now that people will need supplementation, but we may find differently once we kind of analyze the results. But right now it seems like they probably don't need enzymes. But in established type 1, it does seem like there may be people out there who could benefit from enzymes to help with symptoms that we're not catching, because the first thing that clinicians think of when we think of GI dysfunction in type 1 is celiac. And if you don't have celiac, we're kind of like, well, irritable bowel syndrome, so I'm working with a colleague who actually has type 1 diabetes and helped with development of the first kind of artificial pancreas system. And she went undiagnosed with her exocrine dysfunction for about 7 years before she was diagnosed. And now she, really credits enzymes to having a life-changing, symptom relief. So it could be, possible that in people with type 1, we're missing some, but I think in the pre-type 1 period there, it does decline, but I'm not sure that we're going to see levels low enough that they'll need enzymes, but, I may be wrong.
It is interesting, but currently it's not like in standards of care.
No, it's not.
It's not
Yeah.
recommended even as a diagnosed type 1. I know they did have it in sort of the Poseidon study, but that's very interesting that you kind of you're shining a light on it now.
Yeah, so I'm trying to, again, we're trying to build the data to see if we do need some sort of at least screening for people who have symptoms in the standards of care. But I think the data needs to be a little bit more robust before we go.
Oh yeah, definitely. But thanks for unpacking that.
All right, and then in established, so like I mentioned, there is continued decline in pancreas volume, especially in that first year after diagnosis, but then it seems like it sort of levels out, to the best of our knowledge at that point. And then studies are more mixed on function versus duration of diabetes. So there are some showing worse function with longer standing disease and then some that don't. But it does, there is a large decline around the diagnosis of clinical type 1. And then it seems like there may be some continued lower decline after that. So just quickly, there are lots of hypotheses for why this is happening. So developmental or genetic defects are a potential reason. Like I mentioned, the cells come from a common progenitor. There's only one case report of somebody who actually has congenital exocrine hypoplasia with type 1, and so that seems very rare. And abnormalities in first-degree relatives, the studies have been mixed there. So it may play a role, and honestly, there's probably multiple things that contribute to this. So it's probably not just one thing, but this could play a role, and we need more research in that area. The crosstalk deficiencies have a pretty good case for them. So type 2 does have reductions in exocrine size and function. And it has been shown that some loss of the pancreas volume can be reversed with disease remission. So that's very interesting. But it is more mild, it seems like, than in type 1. There's greater atrophy in the pancreas body and tail, where there's a greater proportion of beta cells. And decreased alpha cell mass also correlates with reduction in pancreas weight. However, it does seem like there is a reduction in acinar cell number in a significant proportion of people, which doesn't really go with the atrophy hypothesis as much. And then there are abnormalities, again, in first-degree relatives and autoantibody positive subjects who have normal glucoses. So there's probably something else also playing a role. One thing that I'm looking at that we're not going to quite get into because of time are the possibility of some exocrine autoimmunity happening. So there are antibodies present in 20 to 70% of type 1 subjects to the exocrine pancreas. And it is associated with reduced exocrine function and also more common in first-degree relatives. And so it's an interesting area that has not been explored very much and I think could use some more research. So we're working there too. Biomarkers, MacKenzie's going to talk quite a bit about serum, trypsinogen, and lipase, so I'll skip over that one. Pancreas volume is something that we're very interested in here. And we have an ongoing longitudinal study within TrialNet of 144 autoantibody negative, first-degree relative, and at-risk individuals, single autoantibody positive, which is another area that could use quite a bit of research, and multiple antibody positive. And this is where we did a baseline and 1 year OGTT, A1C, serum markers like trypsinogen, lipase, stool marker, fecal elastase, and an MRI of the pancreas. And we're seeing, again, the sort of natural history in this other population. And then we also are doing GWAS studies to look at genetic determinants of pancreas size. So more evaluating that, genetic and birth risk, basically. So that study is pretty close to being done as well. I think we have maybe a couple more subjects who need to do their 12-month follow-up, and then we'll be analyzing results from that very soon too. So lots of cool results that will be coming out soon.
Fantastic.
Yeah, and then this is just an interesting study that I recently published that actually showed that in people treated with ATG and GCSF in new onset type 1 diabetes, my hypothesis was that amylase, lipase, and trypsin would be preserved in these individuals because the natural history of these serum markers are that they do slightly increase over time up until at least age 30 or so. And so, you know, in type 1, there about 30% of people have low serum amylase and lipase and trypsin. And so I thought if we treated with an immune therapy, that it could preserve the levels of these enzymes. And that was the case in lipase and trypsin. And so responders to ATG and GCSF, not necessarily everyone who was treated, but those who responded to the therapy did have preserved lipase. 6-month levels are about 115% of baseline versus 80 to 90% in placebo and non-responders. And then responders also had preserved trypsin levels, 115% of baseline versus 90 to 91%. This was a really small study, so it could definitely use some replication, but another interesting sort of indicator that maybe we could be utilizing exocrine markers a bit more in type 1 to either indicate response to treatment or kind of identify different subsets of people, you know, who could use different treatments or sort of when we're doing more individualized therapies. So my conclusions are that exocrine pancreas volume, histopathology, and function are all impacted in pre-type 1. And exocrine inflammation, we didn't talk about it a whole lot, but in that one unpublished study that I showed, there actually was not any pancreas volume loss in those individuals yet, but they did have signs of exocrine inflammation. So that may precede volume loss. Exocrine markers are less than the reference range in about a third of type 1 patients. And changes in volume and function may be useful biomarkers of either progression or response to therapies. And we need a lot more work in all of these areas. All right, I'll turn it over to MacKenzie.
Thank you so much. Fascinating.
Okay, yes, I am MacKenzie Williams, and as Monica said, I'm in Clive Wasserfall's lab, and I'm going to be presenting on modeling exocrine pancreatic enzymes for type 1 diabetes risk stratification. So because Brittany already gave us a nice intro, I don't really have to explain why I studied the exocrine pancreas, but we'll cite the same paper that basically shows that we've known that there have been aberrations in pancreas size in individuals with diabetes for at least 100 years. But of course, as Mark mentioned, we didn't make a distinction between type 1 and type 2 back then. So it kind of got maybe lost in some weeds there. And then in the 70s and 80s, we found that there are reduced levels of exocrine pancreatic enzymes in juvenile onset diabetes, or what we would call type 1 diabetes today. And then also, as Brittany mentioned, there were some recent studies done by Martha Campbell-Thompson and others here at UF, and she showed that there's a reduced pancreas volume in first-degree relatives of people with type 1 diabetes, and also in those with positivity for islet autoantibodies. And to us, this points to a potential role of the exocrine compartment in the earlier stages of type 1 diabetes. And so we asked whether circulating exocrine pancreatic enzymes could inform T1D risk assessment. So for this, I'm going to be reviewing some results from a publication that just came out a couple months ago. And for this, what was already known was that there are altered serum levels of these pancreatic enzymes, specifically amylase, lipase, and trypsinogen, but also insulin-like growth factors like IGF-1 and IGF-2, and Melanie Shapiro and Todd Brusko's lab showed this for her PhD work, and also adipokines. So these have all been associated with type 1 diabetes in some way, but to our knowledge, their utility in T1D risk assessment has not been collectively assessed. And so that's what we did for this paper. And so specifically, we actually didn't do any recruitment for this study. We just turned to the UFDI Biobank, and we selected donors who were either autoantibody negative, and these are our controls, people who were single or multiple autoantibody positive, and then those who have been diagnosed with T1D within 1 year. So we had 4 different groups, and we measured all of the different levels of these markers here. And in addition to that, we also have the genetic risk score for these individuals, which we also included in these models. And we worked closely with our biostatistician, Rhonda Backer, who found a way to essentially use this cross-sectional data set to help us form these prediction models. And so to do that, She used random forest modeling, and specifically, she did 4 different random forest models here. And to sort of get at the whether or not these markers can help us stratify risk for type 1 diabetes, we sort of have these spanning across the natural history of the disease. So first we have random forest 1 or RF1. And this model is trying to find markers that can help us to differentiate between autoantibody negative people and those who are single autoantibody positive. In RF2, we're trying to find markers that can help differentiate the controls from the multiple autoantibody positive subjects. And RF3, we're trying to find markers that can help us tell apart single from multiple autoantibody positive people. And finally, RF4, we're trying to find markers that can help us differentiate between the multiple autoantibody positive individuals and those with T1D. And so just like any machine learning algorithms, we reserve a portion of the data set for training, and these models generate thousands of decision trees. Each decision tree gets a vote, and it says essentially what it thinks the most important markers are. And then those votes are aggregated and we form our models. And from there, we use the testing set to do things like determine model accuracy, to see if we can even trust these results at all. But because random forest models, they can only tell us that a marker is informative. It can't tell us how a marker is informative. So we had to use logistic regression to be able to calculate things like odds ratios. And so ultimately what we found was, first of all, we did not find a single marker that could help us tell apart controls from single autoantibody positive individuals, and that includes genetics. So that was, it is what it is. It's not that weird, but it was a bit disappointing. But what's a bit more encouraging is that in trying to differentiate between the controls from the people with multiple autoantibodies, 3 different variables came up as informative, and that would be lipase, GRS2, and age. And specifically, reduced levels of lipase, increased genetic risk, and younger age tended to favor a multiple autoantibody classification. So moving on to RF3, here we're trying to tell apart the single positives from the multiple autoantibody positives. And for this, actually, the autoantibody specificities themselves were the most informative. So this was particularly the case for ZNT8, but also for GAD and insulin autoantibodies. So if a person was positive for any one of these, they tended to be classified as multiple autoantibody positive. But of course, we already know that these are great markers. They're used for staging, so this didn't really surprise us. But what kind of surprised me was when we move into RF4. So in trying to differentiate the multiple autoantibody positive people from those with type 1 diabetes, both trypsinogen and lipase shook out as being informative. But interestingly, it was reduced levels of trypsinogen and increased levels of lipase that tended to favor type 1 diabetes classification. So right away, we were like, why? This is strange. What's going on here? So we drilled down on this. And as it turns out, of all the 4 groups, the people with the absolute lowest trypsinogen levels were people with type 1 diabetes, stage 3. But the people with the lowest lipase levels were actually this group right here. So the multiple autoantibody positive people. So of course, when the algorithm is trying to differentiate these 2 groups, it looks like higher lipase is pushing towards stage 3 T1D. And so this was surprising to me, and it kind of makes me think that lipase appears to be a bit more informative, perhaps earlier in the disease process, whereas trypsinogen seems to emerge a bit later, closer to onset. And so unfortunately, because this is a cross-sectional study, we were not able to ascertain the positive predictive value of these markers because that would require a longitudinal cohort. And also we could not assess the generalizability of the models because that would require an independent cohort. But lucky for us, we recently received funding to participate in 2 longitudinal studies. So the first one's ENDIA, which Brittany touched on. This is environmental determinants of islet autoimmunity, out of Australia. And for this, the cohort is 108 children that they're actually followed since birth. Every few months, they have blood and stool samples taken until they seroconvert, and then up to a year after seroconversion. And so half of these individuals were seroconverters, and the other half are matched controls who did not yet seroconvert, which is to say they might in the future, but they haven't yet. And this is in collaboration with Dr. Jennifer Cooper. She's going to be measuring lipase and fecal elastase. And then here we're going to be measuring trypsinogen, IGF-1, and IGF-2. And we hypothesize that these analytes will begin to decline near autoantibody seroconversion. So we'll see how that shakes out. And the second study is the Key Questions 1, or the KQ1 serum study. The PIs for this are Dirk Homan and Kate Speak, but this is actually a multi-PI initiative, and we're just one of the groups participating in this. And so the cohort is 178 TrialNet Pathway to Prevention study participants. So these people actually are all autoantibody positive, and we're going to be comparing the progressors to stage 3 versus the non-progressors. And we're going to be measuring trypsinogen and IGF-1, and you're probably like, why aren't you measuring lipase? And the reason is because someone else, another PI, actually is doing Olink, which includes a lipase measurement. So we don't have to do that, luckily. But we're going to be integrating all of these results into one giant analysis. And our overarching hypothesis is that the development of clinical T1D is preceded by defined alterations in the abundance of some combinations of these soluble factors. So if we think about kind of the totality of these two longitudinal studies, what's really nice about them is that NDIA covers us from birth to seroconversion, and then KQ1 picks up at seroconversion and it takes us all the way to stage 3. But these are pretty much in their infancy right now. We actually just ran our first batch of samples for NDIA. We don't yet have the samples in hand for the KQ1. I think it's just a really big initiative, so they're working through all the red tape involved with that. But we look forward to sharing the results of these as they come in. So I'll hand it over to Maigan, but I just wanted to quickly thank everyone involved with these studies, especially Ramsey Grace, because she measured the majority of these samples by herself, and also Dr. Rhonda Backer, our biostatistician, and the patients and their families, of course.
Thank you very much, MacKenzie. I did want to just have a shout out. Did I miss this? Where is Jennifer Cooper situated? Is she in
She's
Australia?
in Adelaide. Yes.
Yeah.
Adelaide,
Okay.
Australia.
Great. Yeah, India is a great initiative based out of there.
We're excited to be a part of it for sure.
Yeah, super. Very cool. Okay, let's hear from the other Dr. Brusko, Maigan Brusko.
As I'm well known. Thanks for having me.
Great.
So I was told to talk about whole pancreas, and I thought maybe I would talk about the whole body instead. So I'm here to say that understanding organ-specific immune responses in the case of autoimmunity for type 1 diabetes, the pancreas really necessitates a system-wide approach, because we know that the immune cells that cause this disease are born in the bone marrow, they're matured in the thymus, and then they traffic to the periphery through the blood. And so we collect blood clinically, and this is going to be our most clinically relevant, accessible specimen that we have. And we have the benefit at UF of having access to these immune tissues so that we can understand once these immune cells traffic where they are going. If they go through the spleen and the lymph nodes, they go back many times. If they go to an organ, sometimes they stay and maintain there, and their job there is to keep the organ cleaned up, to maintain homeostasis, and to be ready to act in times of inflammation. And so looking at all of these organs together is kind of a focus that I have been doing in the pediatric space. And so I thought I would bring it here to talk about a little bit in the context of analysis of even more data than you can think of. So I'm going to start in talking about some data generated by Miguel Medina Cervas and Todd Brusko's lab. And I work with him very closely as well, talking about transcriptional profiling with spatial transcriptomics. To me, the whole pancreas means the pancreas and its associated lymph nodes. So I'll be talking first about some data from the lymph nodes there. What we know about transcriptomics in general is that profiling of single cell populations is really important to help us understand what cell types are involved in the pathogenesis of type 1 diabetes. And a lot of that work has been done in peripheral blood as well as in tissues here at the UFDI and in other locations. But when we go to the tissues, understanding where the cells are in their spatial context really tells us what they're doing, because they can have a phenotype, but they're going to enact their phenotype within a tissue. And so it'll tell us what tissue microenvironment they were recently experiencing in situ. And then in the blood, we can hope to relate to phenotypes that we can see when the cells leave that environment. Standard assays like in situ hybridization or immunohistochemistry are limited to fewer molecules. So when we do spatial transcriptomics, we're able to get a really very large picture of cellular behavior, and then link them to their features and their position within the tissue. So spatial genomics helps us maintain that positional information while telling us what is different about each of the cells that are present within the tissue, as we often have a population of very similar cells that have minor differences based on their location. Like in the case of encephalitis, where a T cell may be present in the pancreas, but we only call it encephalitis when it's within the islet. So to look at the cohort that Miguel worked with initially in the spatial transcriptomics study is we got donor sections from HPAP, the Human Pancreas Program. And we have 5 non-diabetics, 5 autoantibody positive subjects, and 6 type 1 diabetic subjects. And we were able to get pancreatic lymph nodes as well as matched pancreas specimens from these donors and perform the Visium spatial gene expression assay from 10x Genomics. And so this assay actually has a 55 micron capture spot size, which picks up everything that is within that capture spot. And so it's a multicellular resolution assay, so that each spot may contain a few cells that you get information from, but then we have analytic approaches that allow us to take those spots and understand the gene expression within, and then also deconvolute to individual cells that are likely present within those spots. So to start, we have to normalize all of the gene expression counts within the different donors. And so the way these donors are arranged is from non-diabetic to diabetic, and the duration of disease continues left to right. We then look at each capture spot and we say, what part of the tissue is this capture spot within? So here, this is a lymph node. We have stromal, medulla, cortical sinus, T cell zone, and lymphoid follicles identified within a lymph node. So those major regions of the tissue, cells that are present within those regions have specific functions so we can understand what they're doing. And then we can do differential gene expression testing between type 1 diabetic subjects, autoantibody positive subjects, and non-diabetic controls to understand how the immune cells are changing in the pancreatic lymph nodes in type 1 diabetes. So what was most interesting was, of course, the most significant result that we saw in the lymphoid follicle in type 1 diabetes is that lymphotoxin beta is significantly upregulated in the lymphoid follicle in type 1 diabetic subjects. And that means that that is a really interesting pathway target for us to look at in type 1. And the good news is that etanercept is in this family pathway, and that drug is currently going to be trialed for type 1 diabetes. Another thing we found when looking at the pancreas is that there's kind of a chronic inflammatory signature in the type 1 pancreas that we see. And looking at the top, this is regional annotation of one donor, just showing that we can see the exocrine, endocrine, and stromal compartments of the pancreas, pardon me, and identify each of those features well. But when we look at some of the gene expression, we actually see across multiple compartments, some of these genes are just generally upregulated. There's inflammatory genes, and you can see that in the upregulation is shown in red in the type 1 diabetic subjects, that we see interleukin 32, superoxide dismutase, complement, and olfactomedin 4, which is a marker of neutrophils and other immune cells, all present in type 1 diabetes and not really so much present in any of the other subjects. Of course, our multiple autoantibody positive donors here did not have encephalitis, so we wouldn't be able to necessarily look at the cell-specific signatures here. But what you can see is that the pancreas has this chronic inflammatory signature. These regenerating genes are often upregulated in pancreatitis. So what the type 1 pancreas looks like to me in this study is kind of like a chronic wound. There is just an inflamed kind of continual state that is present at onset and that continues for a longer duration of diabetes. So we moved on to a higher resolution assay called Xenium. And so rather than that 55 micron capture spot size, we're now moving to single cellular multi-omic studies where we are using a spatial transcriptomic instrument and a spatial proteomics instrument doing immunofluorescence and understanding the all of these immune markers across the pancreas, as well as pancreatic markers for the exocrine pancreas, because the whole tissue is assessed for a panel of 5,000 genes at the same time. And then we mark back to the tissue locations with the immunofluorescence. In the context of type 1 diabetes, this study was specifically designed to address donors across early to midlife, so that we can understand kind of the differences in disease across a broad age spectrum. This is ongoing, so I can present some of Miguel's early data here. I mentioned that we're doing the Xenium 5K tissue panel, which includes 5,000 genes. We've also added on 100 custom genes, which encompass a lot of the type 1 diabetes genetic risk, which will allow us to add that to the analysis. We're working on cellular segmentation, which is a really critical part of this. If we really want to understand the expression in single cells, we're working in an organ that has a high expression of RNases. It's just a really digestive mess. It's probably the most difficult tissue to assess. So we want to make sure we're looking within each cell and making sure our data are really clean so that we don't get erroneous results where we think we have, a new cell type that is an endocrine exocrine cell or something like that because they're close to each other. So we're doing a lot of work on the front end to make sure that we are looking at individual cells and really clean signal. Adding on to that, we're doing the fluorescence afterwards so we can then make sure we're within islets. We can also then see if we're near islets or if we're near structures within the pancreas with all of these segmentation approaches. So when we look at this, obviously we had a gene in mind to take a look at when we looked at some of our first Xenium assays, and we can see that there are lymphotoxin beta positive lymphocytes present within insulitic lesions. So prior to this, we'd seen them in the lymph nodes. Now we see within the red and green glucagon and insulin positive islets that we can see the T cells. And so those T cells and lymphocytes are present as little dots, and I cannot see. Just have to look with that. The little dots are the lymphocytes. I turned the video off for a second, trying to move the screen so that we can
we see it. It's okay.
Yeah. All right. Well, I can't read my legend, so you can see which color everything is, and I don't necessarily remember. There are T cells present within the islet that are positive for lymphotoxin beta. And you can see the cellular segmentation I was talking about overlaid on top, so we can see each individual cell and where those gene counts are within the cells. And the arrows pointing out those T cells in case the lymphotoxin beta is a little blue dot and it's on the background of blue, so it can be a little difficult to see. And then same thing in another islet that we have cells present, the segmentation, and pointing out that we see both CD8 T cells and B cells that are present with lymphotoxin beta around the islet. I also find that there's heterogeneous lymphotoxin beta expression among insulitic CD8 T cells. So that's not the only actor present in CD8 T cells that are present in insulitis, but it is an important one. So we're very happy to see these cells showing up in multiple donors as well. So what that tells us is we have a target. There's already a therapy going on, so we're really excited to see the results of that trial and to continue this work here to understand how this disease may look different in younger donors who've developed type 1 and older donors who've developed type 1, as well as controls in terms of the whole pancreas and its immune environment in the context of type 1 diabetes and health. This immunofluorescence imaging afterwards is performed on the same section, which is really important to note. We don't actually have to take a subsequent section. We can really map directly back to the location within the tissue. And then working with the current data set, Miguel has gone through and identified the gene expression patterns of the cells and used reference mapping and a lot of literature searching to start annotating the cell types that are present within the data so that we can start doing these differential expression at the cellular level, at the cellular population level, rather than worrying about more global expression level changes. So we can see which cells might be participating. And so we're able to see a lot of acinar cells, of course, in the pancreas and endocrine cells and ductal cells, but we can also see that there's multiple populations of myeloid cells, macrophages within the tissue that we're interested in looking at. And then plenty of lymphocyte populations, as well as some things we don't typically see when we do single cell assays like nervous tissue. And so we can really look at a whole composite picture of the immune, stromal, structural environment and the functional environment of the pancreas at the same time. We're really excited about this study, but obviously these data this large presents a challenge. Putting more tissue through and bigger tissue area gives us a lot of data. So we've got a lot of approaches to try to deal with these data. These large scale discovery efforts really require expertise in adequate tissue preservation, which we have here at UF in spades. Quality assurance and control at every level. We do really hard work on making sure these specimens are good for this assay, validated parameters. So we have to have a lot of knowledge in terms of what parameters we should be looking at to aid in our unsupervised parameters that we've added on. Domain knowledge here helps with that. And then robust computational tools. So on the right is literally just a visual display of the image converting to a UMAP so that you can see how we group the cells and compare it to the real data, but then we can look at each cell individually. So I'm going to talk a little bit about some of the computational tools developed in my lab for image data. So this program is called Kintsugi, which is based on Japanese joinery of repairing broken objects with gold. And so we came up with a bunch of words that suit that acronym. So knowledge integration with new technologies, simplified user-guided image processing. And this is work by Justin Smith, who works with me. He has a lot of emphasis on doing code that is open source and free. So what he did not like about working with initial codex data is he didn't know what was going on behind the scenes when the data came out of the instrument. So he actually went back through and recreated the functions of the codex auto-processing and made it faster, customizable, and allowed us to subtract autofluorescence, which is really important in the cellular segmentation process I mentioned, which allows us to have cleaner data and prettier pictures. So I can show that to you here. So actually this overlaying now is the artifactual data. So there's a really high artifact and a dim signal for collagen here. This is a spleen. And you can see as the curtain moves back and forth that we're able to remove that high signal, pull out the low signal, and show the overall collagen meshwork that's present within the spleen, supporting between follicles and allowing that spongy, blood-filled bag of immune cells to do its job. Again, the notes on Kintsugi are that it's free, customizable, the processing is fast, we can subtract autofluorescence, we have some segmentation add-ons that allow you to segment cells as well as the area surrounding cells, and that publication will be coming out hopefully in the next couple of weeks in bioRxiv. But for me, what I like the most about this is how pretty the pictures come out when it's done. And so I have one of my favorite spleen images here. And again, I just showed you the collagen meshwork here, but now that you can see with CD8, which actually lines these venous sinusoids within the spleen, you can see how collagen actually wraps around and supports that structure. And when we can select individual structures and put cells within that structure, we can also understand where they are, what they're doing, and how they might be involved in various processes throughout multiple organs. So I thought, being the last person, I should wrap up about what we talked about overall today at the UFDI. So we really approach type 1 diabetes from every angle that we can think of here. With a combination of genetics and serology and other information, we can really stratify individuals by their disease risk, which helps us understand how to bin them for studies and compare them. With the extensive pathological annotation that Martha Campbell-Thompson has done in all these NPOD cases and the robust computational tools that are available from others and that we are developing, we can really deeply characterize the pancreatic tissue, including the immune cell presence, which is obviously my favorite, as well as all of the other tissue components and phenotypes throughout in both health and disease. And that relating these demographic factors and immunophenotypes directly to observations of the pancreas and the autoimmune lesion allows us to identify targetable pathways for therapies, which is really important in terms of discovery work.
Fantastic.
Thank you. Stop sharing.
Thank you so much for, that summary slide, as well as all of your work was really phenomenal. We're at the top of the hour, but I did have two quick questions for you, Mark. And it's just sort of a shout out. I wanted to just say that in June 2021, Mark Atkinson, together with Linda D'Amelio and Jessica Den, put together paper that came out in Diabetes Care that was titled "The Women's Leadership Gap in Diabetes: A Call for Equity and Excellence." And I'd like to just say that, you know, at University of Florida, this Diabetes Institute, women scientists are so well represented, so articulate, and doing such an amazing work. It does feel as if, Mark, you know, had that in mind, you know, and assisted in getting that moving in the right direction. So I just wanted to give you a little bit of a shout out there.
Well,
And
no shout out is needed, but I appreciate it. But I'd like you to recognize the people here, because I think what makes UF different has always been we kept the patient and their family members is the main thing. We've been generational. A lot of our trainees have eventually moved on and progressed, because it is an amazing atmosphere that we have here, because again, we work like all of those on the Zoom here to try and see a day where, you know, we can prevent and cure this disease. And so it's been a pleasure to hear today all of this progress. And we value the role you play in terms of getting the word out. So thank you again very much.
oh yeah, no, we do what we can. We assist the innovators, and that's what you guys are there at University of Florida Diabetes Institute. I guess my very last question is, you've got so many great ideas coming out of University of Florida, so many great new protocols, approaches, sort of, you know, can you share how strong is your pipeline between sort of the bench to industry at UFDI? We didn't touch on that, and I know it is fairly strong, and I know a lot of biotech has and is currently coming out of there. So if you could just kind of just do like a couple of sentences on it.
Sure. So I see Todd's still on the line here, and I'd like to defer. I'll give him a second to jump on here. I think that one of the other things that we're very encouraged on is we've tried to break down walls between academia, academic medicine, if you would, and industry, and the lay community and funders, because it's an old worn out phrase, but it does take a village. And we really, really would rather work from an area of siloed. Mike Haller mentioned it this morning. We want to work with everybody and share data, share information to try and accelerate efforts. Todd, what would you add on to that?
Yeah, I think you're spot on, Mark. I mean, I think the big thing is that we know that it takes not only us as academics to do discovery work, but then to have sort of the mid-tier, these early stage companies come out, take a risk, build a lead, and then ultimately have that transition into one of the larger pharma to take it over and really get it into the clinic and do the marketing. So I think you have to sort of have all those pieces and those people working together as a group, and we're willing to communicate amongst all of them.
All right, well, you've heard it here today. We sort of shown a spotlight here on UFDI, and I think people will take away the notion that this is an incredible place to work as a scientist or a clinician, or both. It's an incredibly vibrant community and very forward-looking, and they've got access to some of the most cutting-edge tools. And as we just tapped on, you know, they're very aware of sort of the pipelines to industry. So thank you again. We really, you're one of our favorites, and we champion you, and can't wait to see the flurry of papers coming out next.
All right, thank
Have
you.
a great rest of your day. Bye-bye.
Bye-bye.
You.